Hi everyone, thank you for joining our R4Bioinfo series, titled Transcriptomics. It will be an introduction about bulk and single-cell RNA-seq. So thank you, Maria and Stefano, for accepting our invitation and for being with us today. I'm Hedjat Nani. I am a postdoc at Pasteur Institute of Tunis and co-founder of Our Ladies Tunis. So before introducing our speakers, we will introduce you to the Our Ladies Tunis and the Our Ladies Global Initiative. So please, Monet, can you share your screen?
Yes, for sure. Thank you. Oh, thank God.
Hello and welcome to this workshop session. Before starting the workshop, we'll introduce you to the Our Ladies Global and Our Ladies Tunis initiative. The R community suffers from an underrepresentation of minority genders in every role and area of participation, whether as leaders, package developers, conference speakers, conference participants, educators, or users. As a diversity initiative, the mission of Our Ladies is to achieve proportionate representation by encouraging, inspiring, and empowering people of genders currently underrepresented in the R community and to facilitate individual and collective progress worldwide. Gabriela de Caros founded Our Ladies on October 1, 2012. Our Ladies Global was born and the grant was awarded in September 2016. Since then Our Ladies has grown to 170 chapters in 44 countries and 39,000 members. Our Ladies Tunis is part of Our Ladies Initiative and was created in May 2020 by women working as data scientists and biostatisticians and bioinformaticians. Our goal is to create an R community in Tunis and empower underrepresented genders in the R community. This is the core team of Our Ladies Tunis. Come and join our community on social media. This presentation was made with R Markdown. Thank you for watching this video and enjoy our workshop.
Okay, thank you for your attention.
Thank you, Monet. So today I have the great pleasure to have with me two great bioinformaticians, Dr. Stefano Mangiola and Dr. Maria Doyle. Thank you, as I said, for accepting our invitation. So Dr. Stefano Mangiola is currently a postdoctoral researcher in the laboratory of Professor Tony Papenfuss. His background spans from biotechnology to bioinformatics and biostatistics. His research focuses on prostate and breast tumor microenvironment, the development of statistical model and the analysis of RNA sequencing data and data analysis and visualization interfaces. Dr. Maria Doyle is the application and training specialist for research computing at the Peter MacCallum Cancer Centre in Melbourne, Australia. She has a PhD in molecular biology and currently works in bioinformatics and data science, education and training. She is passionate about supporting researcher, reproducible research, open source and tidy data. Thank you very much, and the screen is yours.
Thanks, Hedia, and welcome everyone to our Tidy Transcriptomics workshop. I'm going to start by sharing my screen. And okay, great. So we have created a website for this workshop. So if Stefano can pop the link in the chat for anybody that doesn't already have it, that would be great. And I think a lot of you would have got this link from Hedia. And this workshop material will remain available after today, so you can come back and have a look at it in your own time as well. So I'm going to start with a little bit of background and just let you know that today's schedule is going to be about an hour on bulk RNA sequencing, taught by myself, and then the second part will be on single cell RNA sequencing, and Stefano will take you through that part. and I'll be running code. And if you've installed the package, the workshop package, you can follow along if you like. Otherwise, you can just watch and listen and maybe try it out afterwards. And if you want to ask any questions, please do. You can use the Zoom chat, and Stefano can let me know if there's any questions there as we work through the workshop. And if we have time at the end, we'll take questions then as well. Okay, so we know from the chat already today that there's quite a number of you that don't have experience with transcriptomics, and that is absolutely fine. So we have a couple of slides here just to very briefly give you a little intro into transcriptomics and what it is. So the transcriptome is all the RNA transcripts that the cell makes. And the transcriptome is a way that the information in the genome, so in the DNA, can get converted into proteins that then function in the cell. And we also have transcripts that don't get converted into proteins but have their own functions, and we call those non-coding transcripts. And so the transcriptome plays a very important role in the cells of all our bodies and of all organisms. And why do we study transcriptomics? It's because it allows us to measure changes in expression of thousands of genes, so we can get a full picture of what's happening within cells. And in transcriptomic analyses, a typical or common experimental design that you might have is shown here. So that's where you might be comparing treated samples, so treated with maybe a drug of interest, or you've induced a mutation, and you're comparing them to some controls, to some untreated cells, and you want to know what are the genes that are changing. So which genes have higher expression with the treatment and which have lower. And so we're measuring these mRNA molecules. And then how does transcriptomics actually work in the lab? So in the lab, we take the samples containing the mRNA sequences, and they get what's called fragmented and converted into this, what we call a sequence library. And that then gets put on the sequencing machine. And out from the sequencer, we get what we call these reads. So they're short sequence reads, normally about 50 to 100, 150 nucleotides in length. And this is the raw data that we start with for any RNA sequencing analysis. And there's different types of transcriptomic analyses that you can perform. And by far, I'd say the most common is differential expression. So that's where you're comparing different groups. And that's what we'll be covering in the first part of this workshop. And I'll also be showing you how you can do a cell type composition analysis. So that's if you have tissue samples and you want to see what cell types are present. So if you've got immune cells present, for example, like what proportion are T cells, B cells, macrophages, etc. Okay, and then this figure here shows a typical RNA sequencing analysis pipeline. So that's where we start with the sequence reads from the sequencer. We map them to our reference genome, so maybe human or mouse, depending on what you're studying. We then count how many genes we have, or how many reads we have for each of our genes. And then we proceed to explore the data, perform quality control on it, before we then go ahead and test for differences in expression between our groups, and then on further to pathway analysis or gene enrichment analysis. And in part one of today's workshop, I'll be showing you how you can perform these steps here using this lovely tidy bulk package created by Stefano, who's here today. Okay, and then Stefano will take you in part two through this single cell part. So he'll come back there to that. So, and then just for anyone who's not familiar with tidy data and the tidyverse, because we'll be mentioning those a lot today. So this workshop is showing you how to perform RNA sequencing analysis in a tidy way. So there have been traditional methods that you can use to perform RNA sequencing analysis with packages like EDGAR, LIMA, and that. And traditionally, you use them in, I guess, they don't use tidy data. And so what is tidy data? So that is a concept developed by Hadley Wickham, and he has a paper on it from 2014. And with tidy data provides a standard structure for a data set. In tidy data, every variable forms a column. So like in this diagram here, and every observation is a row, and every cell is a single measurement. And it can enable you to work in a nice, efficient, reproducible, and collaborative way. And there's a link here if you want more information on tidy data. And we'll also be using the tidyverse set of packages in this workshop. And the tidyverse, if you don't know, it's a collection of packages that we can use to tidy, manipulate, and visualize data. So in this workshop, we'll be using many of these like functions. You'll see us using filter. I saw that was already in the Our Ladies intro slides. Select, mutate, and also we'll be doing lots of visualizing with ggplot. So the tidyverse is a really powerful and lovely set of packages to perform your R analyses with. Okay, so now I'm going to move on to the coding part of the workshop. So if you want to follow along with the code, so if you've already been sent the instructions, which I think some of you have, hopefully you're already set up. And if not, you can come back and do this after the workshop. And so I'll just show you as well. So the code I'm about to go to now, the R Markdown, this is the rendered version of it. So it's the exact same code in here on this website that will be available to you after this workshop. Okay, but now I'm going to switch to R. Okay, great. So in today's workshop, you're going to see how you can analyze RNA sequencing data in a tidy way. So using these new packages developed by Stefano. So they're all been developed in the last year or year or two. So tidy bulk, tidy summarized experiment, and tidy single cell experiment. And I'll just show you. So Stefano's been creating these tidy packages, and this is his row, which is called the Tidy Transcriptomics Suite. And this is a roadmap for the development of these packages because they're still being worked on, and there's more to come. So for bulk RNA sequencing, what you'll see us using here today is the tidy summarized experiment package, which is a way to represent bulk RNA sequencing data. You'll see us using tidy bulk package, which is a way to perform an RNA sequencing workflow for bulk RNA sequencing. And then for single cell, in today's workshop, you'll see tidy single cell experiment being used. There is also a tidy scRNA package that Stefano's developed. We just don't have time to cover that today. And what Stefano is working on is then developing a tidy single cell workflow package. So that's to come. Okay. Okay, so now I'm going to get into some of the RNA sequencing analysis. So for this workshop, we're going to start with where we have the counts. So we've got the counts for the genes in this data set, and I'll explain what the data set is in a moment. So first, I'm going to load the libraries. Oh, and just to mention, so if you've got RStudio, there's a little button up here you can press that will give you a little outline of all the headings. So this can be handy for navigating through and seeing where you are in the document. So I'm just going to open that up and leave that there. Okay, and also in this R Markdown document, what I'm going to do is I'm going to use the play button to run the code chunks so you can see what they're producing, and then I'll explain what's happening. So this one here, I'm starting with loading the packages we're going to use in this part. Okay, and then we're going to be making quite a few plots, ggplots. So this is some settings, some customizations for those plots. So I'm going to load that in. Okay, so now tidy summarized experiment. This is one of the new packages I mentioned. And you're going to see now how that works. So the data that we're going to use for this RNA sequencing analysis here is from this airway package. I'm going to load it here. So this is a package that's available online. And this data set comes from a paper by Himes et al. From 2014. And it's got 8 samples from human smooth muscle cells. And the samples are from 4 cell lines. So each cell line has got a sample that's been treated with dexamethasone and a control sample that hasn't been treated. So 8 samples in total. Okay, so if I load that data and have a look, it looks like this. So it's actually what's called a summarized experiment object, which is so some of you might be aware that it's a type of data structure that's very commonly used in by Bioconductor. Bioconductor is the biggest repository of R packages for biological analysis. So this is a popular way they like to represent the data. So as you can see, though, this is not in tidy format. So we cannot use the, say, nice tools we may be familiar with, like dplyr, filter, select, those functions on this data out of the box. So this tidy summarized experiment package enables us to use tidyverse functions on these common summarized experiment objects. So I'll show you now how that works. So if I load the tidy summarized experiment package here and then have a look at the area data set now. Okay, so before it was like this, summarized experiment format. Now it looks like now it's in tidy data format. So it says here it's a summarized experiment tibble extraction. So a tibble is a tidyverse name for a table. Okay, so here I can see I've got 64,000 transcripts, genes in this data set. I've got 8 samples and I'm looking at the counts for these samples. So we can see that in this data set, we've got our gene IDs. So these are our ensemble gene IDs. We've got sample IDs. We've got our counts for each sample. And we've got this column cell, tells us which cell line it's from. And we've also got the dex column here, which tells us whether the sample is a treated or an untreated one. Okay, so that's how tidy summarized experiment can help us nicely see the data in tidy format. Okay, and we can still, with the tidy summarized experiment package, we can still use summarized experiment commands on these objects as well. So this is assays is a summarized experiment function, so we can still use them as normal. Okay, so thanks to tidy summarized experiment, as I mentioned, we can use tidyverse commands. So for example, and I should say here, so you'll see this, in case you're not familiar, this symbol here, the % greater than % is called a pipe. It comes from tidyverse as well. And it's how we can pipe the information from what's on the left of the pipe into what's on the right. So here I'm taking the airway data and filtering it. So filtering it for the treated samples. So dex is the name of the column I'm filtering on. So if I run that, that will filter out just for the treated samples. So now you should see I've only got 4 samples instead of the 8. So we can also use select, another tidyverse command. So select is what we use to choose columns. So I'm going to choose just the sample, the cell line and the dex column, just so you can see how that works. And another useful tidyverse command is mutate. So mutate can be used to create columns. So here, for example, we can use mutate to create shorter IDs for our samples, because some of the plots benefit from having some shorter names in them, so you can see the samples more easily. So here I'm using mutate to strip off this prefix. This prefix is before every ID. So in the column called sample, and I am then going to select, so the new column, so choose the columns, the new column and the original sample column, so you can see what they look like. So let's give it a second. Okay, so mutate has created this new sample name column, and that was the original sample. So now we've shortened those IDs. Okay. Okay, so that's just showing how you can interact with this data set, this summarized experiment object using tidyverse commands. So really, really nicely, thanks to tidy summarized experiment. And now I'm actually going to start getting the data ready for the RNA-seq analysis. So I'll run this and explain. Okay, so here I'm taking our airway data set. I'm creating that column with the shorter sample IDs, explained. And I'm also getting gene symbols. So this is a common thing we do as well. It's because at the moment, I just had the gene IDs, the ensemble gene IDs, but the gene symbols can be more informative later on for knowing what the genes are. So I should see now, I have can I see here, a gene symbol column. Yeah, so a symbol column has been added. We'll see that. Okay. So now we get on to some of the steps for an RNA-seq analysis. So one of the things we commonly do is to remove any genes that don't have many counts across all samples. So this is, we can do this with tidybulk using this keep abundant function. So it's tidybulk I'm using now. And for that, we say which column we want to use to specify our groupings. And that's how it's going to determine how to filter the samples. So if I run this, so that's now, we had about 16,000 or 60 something thousand transcripts, and now we've got 15,000 after filtering out those ones, the genes that are not going to be informative. Okay, and then another step we do in an RNA sequencing analysis is we normalize the counts, or we scale the counts. And this is so we can correct for technical differences between the samples. And these can be differences due to different amounts of sequencing. So they might have different numbers of reads, total reads that have been collected, but we need to correct for that. And also they may have differences in composition between the samples. And I've put a link here where you can get more details on that. And here we can use tidybulk's scale abundance method to perform this scaling of counts for us. And what that's going to do is produce a couple of columns, but this count scaled is what we can see and can use. So if you see, this was our original counts column, and now with the scaling, tidybulk scale abundance has scaled the counts for us. Okay, and because tidybulk works so nicely with the tidyverse, it's really easy to use existing tidyverse functions such as pivot longer to reformat our data that we can then go ahead and plot using ggplot and make nice plots with that. So what I'm doing here is I'm making density plots of the counts for the samples. So both the raw counts, so before the scaling, and also after the scaling, so I can see the effect on the samples. So as you can see here, after the scaling, the distributions are more similar. And from a quality control perspective, we don't have any samples that are looking odd compared to the others. So this looks quite nice. Okay, and for comparison, so if we were going to use a traditional way of analyzing this data, for example, using edgeR in base R to create to do what we just did there, like creating those density plots, we would have to use a lot more code compared to this tidy bulk way of doing it. So if you look here, there's a lot more code involved. So that's the non-tidyverse way. Okay, so we've taken the RNA-seq data, we have filtered out the genes with low counts, we have scaled counts, normalized across the samples. And now we're going to do some more exploring of the data. So extremely common thing we do with RNA sequencing data is to create a PCA plot or an MDS plot to see the groupings of the samples, to see the relationships between them. Do the samples group as we expect? Are we going to see all the treated samples grouping together and the untreated in another group? Are we going to see any outliers, anything odd? So tidy bulk has this function that we can use to do this, create PCA plots, and it's called reduce dimensions. So we give it the scale counts, and it's performed the dimension reduction for us, and it's calculated the first two principal components. And if we have a look at that object that it's created, count scale PCA, so we can see, actually I'll show you in a moment. So it's added these two columns on, PC1 and PC2 for the principal components. So an easier way to see that is to take the object, count scale PCA, and get the sample specific information from it. So I'll show you the difference there. So this enables us to get the PCA information just for the 8 samples. So we've got 8 rows here compared to our output here where we had 127, because in this table we have all the transcript information as well. So here we can see for each sample, if I move along, these are the 2 columns, the PC columns that have been added for principal component 1 and 2. And so we can then use this information to create our PCA plot. So that's what I'm doing here. So taking the PCA information and again, simply piping it into ggplot to make a PCA plot. And so we can see that our treated and our untreated samples do separate. Okay, or another way we can see the relationships between our samples is using heat maps. So our hierarchical clustering. And let's run that there. So we don't want to plot all the data, cluster all the data. We want to extract just the 500 most variable genes, and tidy bulk has this keep variable function. So we can use that to select the top most variable genes and then proceed to create a heat map. And I'll show you that below. So here we've got our 8 samples and we've got them clustering by treated and untreated, except for you can see there's one cell line that's a bit different. So there's one cell line that's more variable than the others. And this is some information that we can then account for when we're going to proceed to do our differential expression. Okay. And again, just to say, tidy bulk enables producing this heat map in not too many lines of code in a tidyverse way compared to if you're going to do it with, say, base R and edge R, so the more traditional way of doing it. Okay, so now we filtered our genes, we've scaled our counts, we've had a look at the data using PCA and hierarchical clustering. Now we're going to go on with the differential expression because we're happy with how the data looks. So tidy bulk has a function to perform the differential expression called test differential abundance. And what you do is you give that the counts object and you give it a formula. So we use these type of formulas with RNA sequencing analysis. So this is saying, I want you to compare the groups to test the differential abundance in this DEX column. So the treated and untreated groups that are contained there. And we're adding in what we call incorporating an additional factor. So this is for the cell line. So we're giving the cell line column as well, because we can see there's some variation due to cell line. And a really cool thing about Tidybulk is that it allows you to run more than one RNA sequencing analysis method if you want, or to compare how different methods work, because there's several very popular RNA sequencing testing methods. So some of you may have heard of them. Like we've got EdgeR, we've got DE-seq2, we've got Limavoom, and it's not always easy to know which one to choose. So thanks to Tidybulk, you could run one or run them all. So this is an example of running them all. I'll show how you can compare results then. So I'm going to set this running. So this is using the EdgeR quasi-likelihood method, EdgeR likelihood ratio method, it's using the Limavoom method, and it's using DE-seq2 method. And so it's using the pipe to run each of those methods and combine the results. And I'll show you what that output looks like in a moment. And it's adding this prefix to the output column so that we know which columns come from which method. Okay, so that's done there. Okay, so you can see we've gotten a whole load of extra columns added, excuse me. So up to here, so we've got EdgeR methods, the two different methods, output columns, we've got our Voom columns, and we've got our DE-seq2. Okay, and a way we can visually compare the results of those methods is we can, so similar to, I was using the pivot sample method before to extract just the sample information from the table. There's also a pivot transcript method where we can extract just the transcript information from the table. And I can use that, I can get the p-values for each of the methods and use this from a package called ggpairs function to then create these plots here. So pairwise comparisons of each of the sets of p-values. Oops. Okay, so looking at this plot, we can see that the two EdgeR methods, so EdgeR QLT and EdgeR LR method, likelihood ratio, are the most similar. As you might expect, they're variations on the same method, EdgeR. Whereas we can see that Voom and DE-seq2 comparison of p-values, they are more different than the others. So we could dig into that further then if we wanted. So that was an example of a way you can run multiple methods with Tidybulk, multiple differential testing methods, or we could just choose to run one. So by default, the test differential abundance method is going to run EdgeR quasi-likelihood. Okay, so that's just run one method. And again, for a comparison, this would be the traditional EdgeR way to do what we've done above. So filtering out the genes, scaling the counts, and performing the differential expression. So another reason for Tidybulk, and in the following the paradigm of tidy data, is to use easy to understand vocabulary. So it uses function names like scale abundance and test differential expression that are more human readable or human intuitive as to what they're doing. So it can be a bit harder for say biologists new to RNA-seq to know what these different functions are doing. Okay, so now to show an example of more visualization so you can do using Tidyverse. So a common thing to do with the results of a differential expression is to create a volcano plot. So I'll create one and show you. So we can take our counts differential expression results and pipe it in to create a scatter plot with ggplot. And this is what we call a volcano plot. And a volcano plot helps us to see. So every dot here is a gene, and the genes towards the top are the more significant. So they've got the smaller p-values for this comparison. And the genes on the right are the ones that are upregulated. So in this case, we're comparing treated to untreated. So it means the ones on the right have higher expression in the treated samples versus the untreated. And the ones on the left of the 0 are the ones that are down. So they're lower expression in the treated samples versus the untreated. And for a volcano plot, we commonly plot, it's the negative log 10 and on the y-axis, and it can make it hard to see what was the original p-value, which is what a lot of biologists think in terms of. So a nice thing about Tidybulk is that it provides this scale that helps you to see what is the actual p-value. Okay, or we can make even more, that's quite a basic volcano plot. We can use the power of Tidyverse and ggplot to customize the plot to our liking. So we can, for example, we can add in the symbols, so the gene symbols for our top most significant genes. So here I'm taking the counts, the differential expression results. I'm getting the transcript table information. I'm sorting by p-value to have the most significant at the top. And then I'm just taking the top 6. So that's these here. So there's our 6 genes there, most significant. And then just to show you how you can take advantage of all ggplots, nice customizations to make nicer volcano plots. So if you look here, so now you can see we've got our volcano plot with our most significant genes labeled, and we have modified the coloring and size of points, etc. Using all standard ggplot2 customizations. Okay, and another cool thing about Tidybulk is that, so it's using other methods under the hood, like you've seen, like EdgeR and that. Tidybulk will actually keep track of the references for the methods you're using. So, and they're attached to your object. So you can then use this get bibliography function in Tidybulk to get all the references for the methods that you have used. So you don't need to worry about what they are or keeping track yourself. And you can take this information, and this is in bibtext format, and pop it into your reference manager, and you have your references there for your analysis that you've performed.
So I have a question about it.
Mm-hmm.
So you can put your references in a bib file or from Mandalay, for example. Can you export them to R Markdown?
So sorry, is the question, can you export?
Yeah, can you export the bibliography from Mandalay to R Markdown?
I would say yes.
Yes.
Because this workshop, actually, if you look at the GitHub, it's got like is it a bibtext file for the references that we're using in the workshop?
Mm-hmm.
So you can definitely create a set of references, so a .bib file, and then in your R Markdown, I'll just show you at the top, you would then point to where that file is. So here, okay. And then you can reference your papers in your R Markdown file. So using the symbols as you'll see us using. So like these @ symbols.
Article. Okay, thank you very much.
Any other questions at this point? Or
question? Yeah.
Because I'm about to just do the last bit, which is on cell type composition analysis. So to move on from differential expression. So if anybody has questions on the differential expression that they want to ask, please feel free to do so. Okay, I'll keep going. I'll move on to cell type composition. Okay, so above I've been showing you how you can compare different groups, so our treated and our untreated, and see the genes and expressions that are changing. So another type of analysis you may want to do if you have, say, tissue samples, is to know what cell types are present. So if you've got different immune cell populations within your samples, different T cells, B cells, etc. So for that one, we're going to use this and subset of a data from a big public breast cancer data set. So it's from the Cancer Genome Atlas. So what we have is patient samples, and we have gene and count information for those. And we can, with using TidyBulk, we can determine the proportions of different cell types within these samples using this TidyBulk deconvolve cellularity function. So what deconvolve cellularity does is it provides access to a number of published methods. So similar to the way test differential abundance above allowed you to use EDGAR, LIMA, VUM, multiple of the popular methods, deconvolve cellularity will allow you to use CyberSort, EPIC, LLSR, and to use those to deconvolve cellularity. So here I'm going to use the default CyberSort method. And I'm going to run that. And this is going to determine what proportions of cell types are present in the samples. And how it's going to do that is it's using this signature called LM22, and it uses that to identify 22 human immune cell types in the samples. Okay, so here we've got the output now, and we can see we've got some information from B cells, plasma cells, T cells, NK cells, and I'll show you a plot with that now in a moment. Okay, so what we can do now is we can take that output, and again, like you've seen, or like we've done before, is we can take advantage of tidyverse functions like pivot longer to reformat that data and to create tidy data format like below. So we can then go ahead and plot this. So what I'm doing here is reformatting it. So I've got a column with the proportions of the different cell types in the samples. And then I'm going to proceed to make a bar chart with this information. So here, now we've got for each of these patients are along the x-axis. We can see what proportions of different cell types are in these samples. So the different cell types listed here. So we can see, for example, that macrophages is quite high in a lot of the patients. So hopefully you can see there, you can do this thanks to TidyBulk. You can determine the proportions of cell types and plot them in a few lines of code in a nice tidyverse way.
We have a question.
Yeah.
So Alejandro is asking, please, can you advise us how to choose the best method to the differential analysis expression? Can you advise us a reference paper for that? Thank you.
Okay, so is this a paper then comparing the different methods on, say, DSeq2, EDGAR, LIMA, VUM? Is that what you mean, those methods?
Yes.
I can't see the chat, it is.
Yeah, he's saying yes.
Oh, okay. Let me think. There's been quite a few comparison papers over the years, and I'll see if I can find a fairly recent one. But the conclusion is with a lot of them is that EDGAR, DSeq2, and LIMVUM are all very good. All very, like, you could choose equally from among them. Some say LIMVUM is fast. So if you've got a lot of samples, like hundreds of samples, that can be a good one to use because it's faster than the others. Stefano, I don't know if you have feelings on how to decide what to choose.
Well, one thing to observe is to compare them, on your analysis, as we have done. So you can see that many of them produce the same results, basically. And you can see for what genes, for example, they might produce different results. There must be a lot of benchmarks out there that compare them, although it doesn't, I don't know anyone by heart. I can say that I published a recent article on the impact of outliers in bulk RNA sequencing analysis. And there is a robust version of likelihood ratio of HR that is among the very popular one, is the one that is more robust in the sense that is less impacted by outliers if you have not so many samples. So if a sample for one gene is very high compared to the others, might impact a lot your statistics. And this HR likelihood ratio robust implementation is what I'm using now, because I found performs very well. So I can pass at least my article in the chat, and if you Google benchmark bulk RNA sequencing differential expression, you will find a lot of literature on that.
Okay, so
Anna is asking, the comparison part is really neat. Can you also run more complex models regarding differential expression? I'm guessing yes.
Yes, so you mean more complicated formulas?
Yeah.
You can, yeah. Isn't that right, Stefano? You can use whatever. You can do multiple contrasts. You can.
Yeah. So, the nice thing about also tidy bulk is that you can just change one argument of the function. You can apply the same model to many models and compare them. Yeah. So, arbitrary models that you can do with the single algorithms you can do with this framework.
Yeah, and I think Stefano's adding in, there's a VUM with sample weights function where you can apply weights to samples that may not be such high quality if you've only got a small number of samples in your data set. So I think that one's on the way as well as an option.
Yeah, it's already there, and also the robust method I'm talking about is already there. So you can do question mark the name of the function, and you have all the list of the possible methods.
Yep. Okay. Any other questions? Because if not, I'll just summarize then this part here. So we have our key points. So hopefully you've seen in this first part how bulk RNA sequencing analysis can be analyzed in a tidy way. And it is just a short workshop, so we haven't had time to go into a lot of the details on RNA sequencing analysis, but there are links throughout the document where you can go back and read in your own time more information on the methods if you like. And hopefully you've seen that tidy summarized experiment, this new package from Stefano, enables you to visualize RNA sequencing summarized experiment objects in a tidy way and to interact with them using tidyverse commands, as well as summarized experiment commands. And I hope you've seen that some of the key steps we perform in an RNA sequencing analysis are filtering transcripts with low expression, scaling for differences across samples, and testing for differential expression, as well as a lot of visualizing the data to see what it looks like. And how tidy bulk package can enable you to perform your RNA sequencing analysis in a tidy way. And it also has some powerful capabilities, such as allowing you to use multiple testing methods, or to keep track of your bibliography, and to also perform cell type composition in your samples. Okay, so that wraps up the first part of the workshop, so the bulk RNA sequencing part. So again, I'm happy to take if there's any more questions. And otherwise, I'll pass over to Stefano.
Yeah, so as many of you do not have experience with transcriptomic analysis, I'm sure you will have many questions in your head. So just type them in the chat.
Yeah.
as we go through a lot of material here. So, you know, we don't imagine that all of you would have everything. So that's why there is a website there. You can go on next month, you know, and use it as a guide if you, I mean, if you enjoy this type of way to do analysis.
And actually, maybe I'll just let you know that if you do have questions beyond the workshop and you want to get in touch with myself and Stefano, you can always submit or add an issue to the workshop website, because we will see that. So if you're on the workshop page, if you click on this GitHub button here, you can go to issues and create a new issue here. So if you want to ask us any question as well, and Stefano and myself will get that.
Yeah.
Okay, get rid of that. Okay, so Stefano, will I give you control then of?
Let's try. Okay, so can you bring me actually to the biological introduction?
Oh, yeah. Book.
Yeah. Here. All right. Cool. All right. So we have touched on bulk RNA sequencing. Now I will tell you a bit of what different there is with single RNA-seq, and as Maria showed, how we can analyze single cell data according with tidy principle that Maria explained very well before. Of course, that's not the only way to visualize data, but as you know, the tidy principle are really taking over the R data analysis community, and they became popular in many of data analysis fields. Transcriptomics is just another example of how we can enjoy this type of analysis. All right, so single cell. Now the difference from single cell and bulk cell in principle is very easy, meaning that while in bulk cell we might take a tissue, we might extract RNA, so basically destroying this tissue, extract RNA from it, and quantify the RNA molecules for each gene. We don't know where those RNA molecules were coming from. Were coming from an epithelial cell, an immune cell, or something else. Okay, with a single cell RNA sequencing, with the microfluidics, we are able to tag every single cell with a specific barcode. Then after doing that, we do our RNA extraction and sequencing, but then we are able to, thanks to this barcode, to identify the transcripts that are coming from the same cell. And normally we might sequence, 7,000 cells, 10,000 cells from one tissue. And for each of those cells, we might be able to detect 3,000 transcripts. And so the analysis we can do are really much more informative and complex, because we can do, for example, differential expression analysis, as Maria told you, but between for each different cell types, and really dive into the biology of the tissue. So just to give you an overview, we have again a kind of standard baseline analysis we can do with single cell data. Of course, we can do much more with this. So there are here a few different steps that are not present in bulk. Again, just briefly, we have a read sequenced, row sequence read. We map them to the genome, and we understand, quantify the RNA abundance for each gene. We do data exploration. For example, we plot the different samples we have in space, so we can understand which sample and which cell type from those samples are similar to each other. we can label, actually we can do cell clustering, meaning that we try to understand for each of those cells the similarities. And once we have identified this cluster, we can try to give a label to them. For example, immune cells, epithelial cells, fibroblasts, and so on, based on their transcriptomics. And then we can do comparative analysis, for example, test differential cellularity, whether a patient might have more immune cells than another. Test, again, differential expression between clusters, lineage trajectory, and so much more. Okay, so this briefly is what we will touch based on. So I'll go back to the workshop and I will introduce to you the package that we'll use today. Okay, so again, there are two main frameworks we can analyze single cell data with. There is SURAT, which is present in CRAN, the biggest, the official repository, R repository. And there is Bioconductor, which is the biggest code repository for biological analysis that has several different packages that interact with each other and complement each other, and they can be used. So in this workshop, we will touch on the Bioconductor repository rather than the SURAT framework. So we have different packages in the Bioconductor repository useful for analyzing single cell data, including scatter and scRN and scuttle. And you can see here other packages that we will use in the future. Actually, Maria, yeah, maybe it's better as you did. I'll go to the RStudio session. So I will execute the codes from here. Okay. As I said, we load these different libraries. Now, as before, some are like for bulk RNA sequencing, Summarize experiment is the, let's say, official data container for bulk RNA sequencing data. And we developed a tidy Summarize experiment to represent and manipulate this data in a tidy way. Now, similarly, there is a single cell experiment, which is the official container of single cell data in Bioconductor. And we developed a tidy single cell experiment, again, which is an adapter to visualize and manipulate single cell data with tidy principles. It works more or less in this, actually works in the same way as I will show you now. So all the tidyverse function that you might have learned are applicable to this object as well. So you virtually do not have to learn anything more for manipulating these objects. So I will, I'm pretty sure this will be visualized as tidy already. So as you can see here, similar structure is a tidy data frame. You can see here that is a single cell experiment table abstraction. We have 51,000 transcripts, and we have assays that is the name for the the container of our actual transcript counts. We have counts and log counts, so counts transform with the logarithm. And the one difference that we have compared to bulk RNA sequencing data is that tidy single cell experiment give us, by default, cell-wise information. So it doesn't give us transcript-wise information. And the reason is that with single cell transcriptomics, we are generally much more interested in the beginning to cell-wise information and analyze cells rather than go straight into observing transcript values and differential expression analysis. And because if we would visualize all data here, this data frame would become huge. Okay, so we have a easy way to extract transcript information. By default, we can see here the cell ID, the file this information was taken from, and few other summary information here on how many, the total counts and the total number of transcript in each cell, and some cell cycle information and so on and so forth. Okay, and again, even if appear to us in a tidy format, is still a single cell experiment object, and so we can query it and analyze it as, you know, if it were a single cell experiment object. And here an example, we'll use this function that works on that kind of object on this tidy representation, and obviously it works. It gives us the assays we have seen before. Okay. Again, an example on how we can subset information, but we could do virtually everything we could do with the tidy data frame with tidyverse. So filtering, manipulating information, joining different data sets, so on and so forth. An example of this is we want to clean up a bit our data set. And so our file column includes some useful information, which is a sample ID and the data set and groups that these samples belong to. And so we can use extract, which is a function from tidy R on our object, extracting this information from the file column, which contains a long file path. And we can see here that if we select a sample dataset and groups that we have just extracted, here they are. Okay. Now, there is a quite complex concept that we are explaining now. I don't know what you think, Maria. I might touch on quality control and keep nesting if, you know, for the end, if someone is interested, as I think is a bit overwhelming at this stage.
Sounds good. Yeah, if there's time, people want to can come back to it. Yeah.
Yeah. As nesting is a bit an advanced concept, as many of you, you know, you can let me know what is your knowledge in tidyverse, and if someone is interested, we can explain it. It's very powerful, but a bit hard to understand if you have never worked with tidyverse. Anyway, let's go to quality control. We use nesting for, but I will focus on quality control. So it's important, as for bulk RNA sequencing, for single cell RNA sequencing, try to understand which what we call cells, for example, are real cells, or the droplets that should have contained cells were empty, and so we were just capturing ambient RNA that was just free in the solution. So we don't want to analyze those because that would be confusing. And also, when we process the tissue, we have to digest with enzymes, and so some cells might die. And in the meanwhile, we are processing might completely change their transcriptome. So also, we want to recognize dying cells and just avoid them. So let's start from one data set. So I will not have to go to nesting necessary. Usually we do quality control for each data set. And so we select one of them with this ID, as we have many samples in this example. So the first thing we can do, we are using common function. This is nothing to do with tidy, is functions from the Bioconductor repository. In this case, for each, we want to detect the location of each gene, because we want to select the genes that are and use the the cells that express a lot of mitochondrial genes to discard them, because it is an indication of cell death. We use these, let's say if I can, increase the, I wanted just to decrease this left window, but it's fine. We use this function per Q cell metrics that it will output a list of statistics about qualities of our cells that I will show you now. We are indicating which genes are from the mitochondrion because we have identified them with this in this location variable. And you can see here that we have a lot of statistics from each cell. This is the cell barcodes. And we have, for example, mitochondrial transcription percent. So we have here 4%. For some of them, we might have 50%, 60%. And other statistics like the sum and the transcripts detected, the transcript abundance here. All right, so now that we have these statistics, we can convert this data set into a table as we are used to interact with tables. And we can use, again, the tidyverse way to do things to create a column with mutate where we label what is for us a high mitochondrial cell, a cell with a lot of mitochondrial transcription. And this, we will do it in a robust way using isOutlier function, again, from Bioconductor, okay? And basically what we are creating is a table similar to before, like cell barcodes, but this time we also have high mitochondrial cells that we will want to discard, okay? Now, we have these data sets that is giving us information for each cell. So we just use another convenient tidyverse function, which is left join. It simply joins our original data sets, which is a tidy single cell experiment, with this normal table using the cell barcode. Okay, so we are not risking to mix up the two tables when we join them together. And you can see here that we have the table we had before, which is a single cell experiment abstraction, with also our new columns, for example, high mitochondrion. Okay. Okay, this part was going into nesting. I will not go now into nesting, but I just can tell you that since we have to perform these quality check per sample, for example, if a sample is coming from a patient, another sample is coming from another patient, a convenient way, instead of splitting up this data set, doing a for loop, rejoining the data set, so on and so forth, there is a powerful concept in tidyverse that is called nesting. That feel free to ask me at the end if you are interested, but the material is here, and we can go back afterwards. Okay, we have seen quality control. We have eliminated the dying cells and also the cells that were not actually cells, the droplets that were not including cells, and now we have high quality data set that we can analyze. As before, there are some samples that can have been sequenced at higher depth, meaning that for one tube containing a solution, we might have produced more information about. And so we really are introducing some technical variability where all transcript for one sample would appear as much more abundant. And obviously this is an artifact. So we want to scale, is also called normalization as a jargon that I'm not in love with. It's just scaling. We want to rescale the data to take into account these sequencing depth, a technical difference. And another thing that we see here that we were not seeing so much in bulk RNA sequencing is integration. Because we have many cells for different samples, And we might have also another source of technical variability because simply because different samples might have gone through different procedure. Maybe one sample was processed one day, another sample another day, and so this might affect the chemistry or many aspects of the processing. And so before we build cell clusters, we want to eliminate as well with this technical variability, and that's called integration. Okay. Okay, so all right, so here we are using nesting. I forgot that the quality control wasn't the only one, but basically I will briefly show what nesting does. For the moment, not going too much into detail, but basically we are again we are creating a batch column for each data set. We are considering each data set as a different batch. And we are separating our data set according to batch. Okay, so I will just show you as the data set will appear when nesting, so you will understand what I'm talking about. I cannot pass. Maria, can you pass these 3 lines of code in the console, in the execution?
It's because you didn't run some of the commands above where data set was created, I think. Okay. Skipping over the...
Can you try to...
run?
Yeah, oh, sorry, I didn't.
Okay, so I'll run these to generate. Oh, so it must be up here creating the data set column.
Might be because we are overriding the...
We do have... This has all worked, everyone. Sorry about this. This is just because we are changing a little bit on the fly here.
I think, yeah, I think the object changed because just we have reassigned to that object. Now it might look different. Maybe we can...
Here, do you want me to... I'll just quickly...
No, because now the object has changed because we are saving space. We are reassigning, but it's fine. Let me print pbmc. I want to see how that it looks now. So I go down where we are at the moment. So, here. Yeah, here you go. You see, changing plans on the fly. But people hold on. Meet, okay. Getting there. Okay. Quality control nesting. Oops. Yeah, that's a better way. All right. So let's see. You see why? Because we have integrated and by conductor function have changed the object. Okay. I will, for the sake of going on, I might show you with as the nesting data set was looking, but I just tell you that here we are separating the different batches and feeding them into this multi-batch norm function, which integrates them and recreating the table. Okay, we are doing this and the object change because these functions are not, especially these fast MNN, which let's say perform the data integration, doesn't run on data frames. Okay, so we are changing basically the structure here. Okay, but basically, again, we have separated the batches, feeding them through this function to prepare the data sets, and then unite them back with this fast MNN. Okay, and this is the object we get back. Again, is represented as table, and we can see here that we have our batch column, and these are the reduced dimension of the data that have been corrected. Okay, and now I will, like Maria showed you before, the PCA, so the principal component analysis that reduces high dimensional spaces, for example, with 20,000 transcripts, so reduces 20,000 dimension to 1, 2, 3, 4. And in this case, we have 5 reduced dimension that are called corrected. So basically, now they can be used for cell clustering and visualization. Okay, sorry for this inconvenience. Now, we have integrated the data, and so all samples can be in a way analyzed together. So the technical variability coming from each samples has been adjusted for. Okay. And now what are we trying to do is to try to identify groups of cells in our data set that have similar transcription and label them as such. And so what are we doing here? Again, is using simply bioconductor methods. So nothing tidy about here. This is a quite common standard analysis. We use build SNN graphs, which is a method that tries to model our data as a graph. Now, the why is a bit complex. If someone is interested, I can explain further. And the clustering algorithm that is called cluster workshop works on this graph that describes similar cells to identify what is called cell communities that are, again, transcriptionally similar. And we extract the membership, so the cluster label. And we add them to the what is called the cell annotation part of our object. So nothing tidy about here, it's just standard analysis. But again, we can visualize the result in a tidy way. And we can see here that our label has been added. So there are, you know, roughly 7 clusters here, and this is our label. Okay, maybe we can, are there questions? We can take.
Is there any question?
Okay, if you have some question, we can take 1 minute break and answer some of them. And even the most basic ones, don't be afraid to put them in the chat. All right. So just to summarize what we have done so far, we have imported the data sets, clean up the data sets, run quality control and filter the low quality cells. We have scaled the data and integrate the data to eliminate technical variability between samples. And we have identified cluster of cells that are transcriptionally similar. Okay. Now, we can, of course, visualize these cells in a space, 2D or 3D space. And for doing this, we can, similarly to PCA, as I mentioned before, reduce the dimensionality of this data. And one way is to do it with PCA, but because the original dimensionality and structure of this data is much bigger, linear dimensionality reduction are not the way to go, like PCA. And so there are non-linear ways, as we can see here, which is UMAP, that resolve much better the local neighborhood of each cell, really giving us the structure, representing to us better the structure of our data. And now, normally we can visualize these UMAP reduced dimensions with common bioconductor functions in 2 dimensions. But just to show how convenient is this tidyverse integration, I will show the use of another kind of tidyverse friendly function, which is plotly, which allows pretty easily to produce interactive graphs. And in this case, we'll produce a 3D graphs of these cells in space. And because we have our reduced dimension column here, we can conveniently feed them directly to this plotly function and color them by our cluster identity. And you can see here, let's see if I can manipulate this. We have largely 4 clusters, 4 large clusters of cells, and each of them contains 1 or more clusters that are visible here with different colors. And so you can appreciate the diversity of the data in this way. A 3D visualization is particularly interesting because with this complex data set, really you can resolve some heterogeneity that sometimes with 2-dimensionality visualization is quite challenging. Okay. So, but this is just an example of how easy you can do quite complex visualization with this tidy single-cell experiment. Okay, so we have clustered the data, reduced the dimension for visualization, and now the next step is each of those cells of cluster of cells which are transcriptionally similar. Basically, what are they? What type of cells? You know, if we sequence blood, there will be mostly immune cells circulating there. If we sequence skin, there will be mostly myeloid epithelial cells and melanocytes and so on and so forth. So we want to understand the composition of this tissue. And for bulk RNA sequencing, we had to use an inference method like CyberSort to try to infer from a mixed data what were the proportion of immune cells, epithelial cells, and so forward. In this single cell data, we can actually have a much more direct observation of this. We have just to classify a cluster of cells. Okay. So,
Stefano, sorry.
Yeah.
Before moving, so what's the difference between the PCA before the reduced dimensions?
Yeah.
The reduced dimensions part, the part.
Yeah.
Yeah. So you said that we can't do PCA when we have like higher dimensionality. So it's better to use UMAP.
Yes, well, let's say PCA is not enough. Actually, UMAP is a dimensionality reduction method that is in the most common workflow run on principal components. So basically, first you summarize the data with PCA, and then on those principal components that have been identified, for example, the first 30, you further reduce the dimension onto using UMAP, which is a non-linear method for dimensionality reduction. So if you visualize the first 2 principal components of a single cell data set, you will find that it's not so informative. Visually, you don't see the different cell clusters. And this because, again, the single cell data set is very highly dimensional, and there is a lot of structure that is hard to capture with the linear method. And so UMAP ultimately allow you to, in 2 dimensions, to really refine the structure of your data. and define local clusters. Okay,
Thank
so
you. We have a
once-
question here. What is UMAP 1, 2, etc.?
Well, the columns that are called UMAP are the columns that include the first dimension of UMAP, the second dimension of UMAP. Similarly to before, we had PC1, which was the first dimension of principal component, PC2, and so on. So UMAP 1 and 2 and 3 are just the first dimension of UMAP, second dimension of UMAP, and third dimension of UMAP.
Thank you.
Are just coordinate in space. Okay, so what about the identity of these clusters? As before, we have reference signatures where we know that some cell types transcribe preferentially some genes rather than others. Those are called marker genes. And we apply this bioconductor function from the scRNA package. It's called find markers. Sorry. Okay, so before diving into this, I should mention that there are two strategies for cell type classification. That's a more manual strategy, a kind of a manual curation of the data, and there is a more automatic cell classification that we will see. In practice, usually we use them together. So we first automatically classify those cell clusters and then manually curate them. So I do both, for example, and I will show you them both here. Let's start from manual classification. What are we doing with manual classification is, first of all, we have 10 cell clusters, let's say, and for each of the cell clusters, we compare one cell cluster with all the others, and we identify the genes that are most highly transcribed in one cluster rather than the others. And again, these are called marker genes. And we can visualize these marker gene datasets that we have produced with, I cannot pass the code. Let me. And basically what we have identified is for each cluster simply a list of genes, okay, that we have put together. So all these genes are marker genes for our clusters. And then we can use heat map to start visualizing which genes are highly transcribed for one cluster and compared to the others. And here we have produced a heat map on the x-axis here. We have cells, so there are a lot of points here. And on the y-axis, we have transcripts, so the marker transcript we have selected. And you can see here that roughly, even without forcing clustering, separation, we have quite good separation of our clusters, as we would expect for a, successful analysis. So this is a good thing to check. And then what we would do is to go to the literature or using the knowledge we already have to saying this HLA gene might be abundantly transcribed in monocytes rather than macrophages, or rather than inflammatory immune cells. Okay, and so we basically go cluster by cluster, analyzing the marker genes for the cluster and annotating what cluster it is. Okay, of course, there are algorithms that have been produced to be able to do this procedure automatically. And so there are a lot of software that can do it, but today we will see just one of them. This is called Single R. It's a quite popular software. I will run you through the code. The principle is, as sorry, I was mentioning before, where Maria was analyzing bulk cell. We have publicly available transcriptomic signatures of known cell types. And we look for them in each of our clusters. And the one that scores better, we attribute as label to our cluster. So if our cluster has, has a high abundance of CD3, which in our here blueprint encode data, which is our reference data frame data set, says that these genes is identifying T cells. The automatic algorithm will say, okay, this cluster is T cells. Of course, we have a lot of marker cells for a lot of marker genes for a lot of cells, so the algorithm is quite more complex than this, but the principle is that we use a reference to label automatically our clusters. And so you can see here that we take our integrated data frame, we feed it to singular, we gave the reference data frame that is from Blueprint, and these are the cell names that are in this reference. And we indicate the cluster identity that we already identified. And then we just parse this data to create a data frame. And you can see here that this procedure has created a 2-column data set with our cluster label that we already knew before. But now we have also a cell type label next to it. And as before, we can join this new table data frame with our original single cell data, simply performing a left join. In this case, we are joining by label, which is our cluster label. Again, just to show you the data, we have the original data set. In this case, a new column called first labels, where we have our cell identity. And usually what happens in practice that you know more or less which cell type each belongs to each cluster. And then if you want to dive better into the biology, so finding smaller, more specific phenotype of these cells, you would use manual curation and dig into the literature and really improve the meaning of your analysis. Okay, so these were all the steps, the most common steps that you would start a single cell analysis with. Just to mention that, for example, using tidyverse function, we can easily create summary of our data. And so here we just counting how many cells there are in each cluster and what identity they have. And you can see here which are the biggest clusters of all. In this case, is skeletal muscles, followed by fibroblasts and NK cells. But again, this is just a small example of many analyses and data manipulation and visualization you can do with single cell data. So this is just for you to get you started. Okay, so I will conclude with a few key points. So some basic steps of single cell RNA sequencing data are, as I shown, dimensionality reduction, cell clustering, and identification. Cell clustering and cell type identification. Tidy single cell experiment is a tidy representation of a single cell experiment object, which is a popular data container in Bioconductor, and they enable us to display the data and manipulate the data in a tidy manner. As we have seen for single cell experiments, similarly, tidy single cell experiment is an abstraction, a tidy abstraction of a single cell experiment object. Again, the most popular container in Bioconductor for single cell data. And it can both be used with the tidyverse functionality, but also, of course, with all methods that work directly on single cell experiments. Okay, so I hope you enjoyed this section. Again, now we can open for question and discussion. We have quite a lot of time, and I hope you interact with us. So don't be afraid to ask questions. Also, if you want to, again, understand better what differences are between bulk and single cell, also really basic questions that have nothing to do with the code, you can also ask.
We have a question here from Anna. Thank you, Anna, for your question. This was a very interesting insight into single-cell RNA-seq analysis. What inspired you both to create the tidy bulk package, and how long did it take you to make it, if I may ask?
Yeah. So, well, the reason why creating this infrastructure that can interface transcriptional data with tidyverse is pretty simple, as most people, even in, among us that analyze genetic data, use tidyverse for analysis, because tidyverse from 5 or even more years ago introduced a much more intuitive and robust way to analyze the data. Now, before tidy bulk and tidy summarized experiment, tidy single cell experiment, you would use tidyverse to analyze your data in general, but you would forget about tidyverse when you were to analyze transcriptomic data. Okay, so you would use BaseR for one thing and tidyverse for another. And of course, it doesn't make much sense. So there is a lot of value in to be able to analyze the data in a tidy manner, even though, even for transcriptomic data. So to bridge these two fields. And this has happened not just for genetics data, but economics, a lot of fields have been bridged to tidyverse way of doing things. And why not transcriptomics? So that's the main reason we have approached. And actually, I knew Maria for this exact reason. So she's an expert in providing workshop for data analysis, and she was doing a workshop of tidyverse and a workshop of RNA sequencing analysis. And it was a bit weird because she was teaching tidyverse for one thing, and then for having to forget about everything of this for the transcriptomic analysis. And she approached me, she was interested, and then we get together and develop this whole framework.
Yeah, exactly.
Yeah, so that's a bit of a story. And how long is, we started last year, and it still is under heavy development, but they are becoming popular now. And so a couple of years, I would say.
Wow, that's great. I saw that in my case, so many single cell analysis are done in Python. Yeah, so what's the difference according to the speed? Because sometimes R is quite slow compared to Python. So do you think, yeah, so can you tell us if, for example, make a comparison between Python and tidy transcriptomic and tidyverse here? Thank you.
Yes, so Maria, feel free to take this if you know more, because I personally don't have unfortunately much experience with Python. I've used it for some other research, but I've been using R for a few years. R can be annoying for many things, especially for performances and stability. The reason why I use R is sometimes convenience is more important, and the kind of ecosystem is more important than performances, although it's always frustrating. I don't hide that. So I don't have an answer to that question. I just know, as you know, that R is not famous for its performances. Maria, do you have experience with Python at all?
I know that, yes, some people use ScanPy is the popular single cell package for Python for single cell analysis, and that's apparently very good. Just like Stefano, more of the people I know still use R, I think because they're using R for other analyses, so they continue to use that for single cell. The scRNA package in R is very popular, and that I think has some functions where it uses Python under the hood, because I think for things like the clustering or the UMAP methods can be quite compute intensive. So I think some of those R functions can actually be using Python, or there are a couple of people I know who do most of their single cell in R, but then might run, say, just the clustering through Python, through using something like the scanpy method for that. So I think in answer to your question, it's a mix. You could.
Yeah, thank you. Yeah, sometimes maybe, yeah, you have to mix, you know, between R and Python. So R is more user-friendly than Python, but then, you know, like the performance, yeah, it's like sometimes it's not.
Yeah.
Thank you. So I don't know if we have more questions.
If you do, yeah, feel free to ask in the chat or even unmute, I guess, at this stage. And I'm also, we have a little short feedback form that I'm going to put the link in the chat to now. And we'd be really grateful if you could spend a minute or two just to give us your feedback on the workshop. Because we, as Stefano said, we've been running a few, so we do take the feedback on board and try to continually improve the workshops we're giving as well. So I'm just going to grab the link for that. And thanks, Alejandro, for your message in the chat. Just getting the link now.
We have a question. Let me see.
Yes, so could you tell us some open RNA sequencing data set we could use to play around with? Yeah, there are definitely. There are very small and popular ones that have not so much biological meaning, but you can still operate on and are quite convenient. And one of them is Pasilla. Airways that we have shown in this workshop, but as well Pasilla. Maria, we haven't shown in this workshop, isn't it?
No, we haven't. Yeah, that's another popular.
Yeah, I mean, I will write in the chat. This is another very, hello world data set. Is a R package. Again, you download Pasilla package and you have the data set with it. For single cell, what have we been using? It's I don't remember if it was taken straight from popular public resources, or we have kind of created unifying different data sets. Let me try to remember. But well, independently from this, if you go to 10x website, which I will write, website, they have several data sets for you to download. That's definitely a way to start. And then will be others that now. Yes, so the feedback is very important. For example, now because I decided to skip the nesting, we had a bit of trouble because the data set was already updated, and so I couldn't show you. So it's important to also to know if what's too much information for you. If anything, Maria prepared this nice questionnaire, and would be nice that all of you would take a few minutes to inform us.
So thank you very much. Thank you for this amazing workshop. Let's see if we have more questions. We have a few minutes more.
I'm just putting a link into a platform I've used to get RNA sequencing data sets as well, if you want counts tables to play around with. I think it's mainly bulk RNA sequencing data sets for human, mouse, and a few others. There are a few repositories out there as well of count data sets that you can use and experiment with.
So I also put in the chat my GitHub repository account where all these packages and workshops are included. And as Maria mentioned before, you can open issues. GitHub is very good for interacting with people. So you can create an issue for any question, feedback, critique, so on and so forth, for every of those packages or the workshop.
How long it takes to analyze single-cell RNA-seq? Like from the moment, you get.
It's, I mean, one thing is computing time, which is, it can go to, many minutes for some steps. But the really big part is the analysis itself. It's very much more complex than one would think. And so once you start to go deep into the data to try to classify cell types, you spend a lot of time redoing the analysis and trying to reclassify the data and so on and so forth. So in general, 6 months for putting up a paper is not a lot. The computation also takes much longer. It could take 15 minutes for some operation to finish or even longer.
So you can run it on your local computer. You don't need a server for that.
I'm not sure about that. Maria, you have experience. I mean, meaning that I would say, no, I don't run on my local computer. But Maria, have you run any serious analysis on your local computer?
No, and I think everyone I know does use a server because it will depend on how many cells you have in your data set. And some of these analyses, depending on the size of the tables you're creating, can be using like even tens of gigs of memory. I've seen up to like 40, 60 gigs of memory. So you're not likely to have that on your computer unless you're very special.
Okay, thank you.
So unless you're working with a tiny data set to play around.
It's very important, information. Thank you very much. Sorry.
No, I mean, even if these objects are highly compressed and optimized, and our tidy abstractions is nothing different from that, it's just a different display of data, those are very big nonetheless.
So even in the RStudio cloud, you can run an a single cell RNA-seq analysis.
Do you mean the public, the free RStudio cloud?
The free,
definitely not, because they have a limit on the free RStudio cloud of, I think it's 1 gig of memory.
Mm-hmm.
So if you pay for like a subscription account, you might be able to, then do it if you can get more memory.
I think if your institute or laboratory or division does not provide kind of a central server, I know many people that, which laboratories or institution invest in some cloud computing like Amazon or virtual machines that you can turn on and turn off when you want, and then you can have decent resources to do analysis.
Okay, thank you very much. So,
and somebody's put in the chat, Alejandro, that you could use cybers if you don't have computational performance. And I know there's some people that use Galaxy, like the public Galaxy servers, for some single cell analyses, because I think they'll have access to more memory.
thank you very much. So I think we don't have more questions. Thank you very much, Maria and Stefano. And thank you for joining us today.
And thank you, Hedia, and the Our Ladies team for the invitation to present today.
It was very nice
for coming along.
From you, yeah, accepting our invitation.
And next time will be physically there.
Yeah, we hope so.
Greece, Spain.
We hope so. Yeah. I hope one day, yeah, you'll come to Tunisia and we can have an amazing workshop.
It would be excellent.
Yeah, but meanwhile, we really enjoyed this, meetup and workshop. Thank you very much. So please stay tuned. We'll have our next R4Bioinfo about tidy genomics. And we'll send you all the teaching materials and all these links in the chat and the recording very soon. Thank you. Thank you very much, and have a very nice day.
Thanks, everyone.
Bye.
Thank you. Bye-bye. Thank you, Batul.